Hello everyone, welcome to BioOcean, and this is me Sheethalmaria with you, and we are here with a new video about the classes of immunoglobulins, otherwise types of antibodies. And in our previous video we have already discussed about the structure and overall idea of what an antibody is, and I have attached it here, and you can get that by clicking the I button. If you haven't watched that still, please do watch and understand the overall concept. And in this video we are going to explain about the 5 different types of antibody that human serum contains. And before getting into the video, if any one of you are new to my channel, please go and subscribe to my channel and press the bell icon so that you can get the notifications whenever I upload a video. Human serum mainly consists about 5 different types of immunoglobulins, namely IgG, IgA, IgM, IgD and IgE. And this classification is mainly based upon type of H chain, otherwise a heavy chain present in the immunoglobulin. Talking about and starting up with IgG, it is the most common and major serum immunoglobulin, constituting about 75 to 80% of the total, and its heavy chain is of gamma. It is a circulating immunoglobulin distributed equally between the intravascular and extravascular compartments. It contains less carbohydrates than other immunoglobulins and has a molecular weight of 1,50,000, and its half-life period is 23 days. Its normal serum concentration is 8 to 6 mg per 100 ml. IgG exists as a monomer and is the only antibody that crosses the placenta, and hence it helps in the natural passive immunity to the fetus. It is a secondary response antibody. After the initial antigenic stimulation, IgM is the first antibody to appear in the circulation. Then it is supplemented by IgG. On the second exposure to the same antigen, the production of IgG is more rapid and the concentration becomes very high, and hence the principal component of secondary response is this IgG. It provides long-term resistance too, and hence it has subclasses of 4 immunoglobulin molecules, namely IgG1, IgG2, IgG3 and IgG4, each possessing a distinct type of gamma chain. Then comes the immunoglobulin A, IgA. It is the second most abundant immunoglobulin and constitutes about 10 to 13% and has a molecular weight of 4 lakh, and hence it has a half-life of 6 to 8 days, and normal serum level concentration is 0.6 to 4.2 mg per 100 ml. IgA is a major immunoglobulin colostrum, tear and saliva. They are found in low levels in serum. It occurs in 2 forms mainly. In the serum, it occurs as a monomer, and in body secretion, it occurs as a dimer and is called as a secretory IgA. IgA is synthesized by the plasma cells situated near the mucosa, and a secretory component or a secretory piece is present, hence it gets the name secretory IgA. This piece is produced by the mucosal or glandular epithelium and not by the B cell. It helps the immunoglobulin to cross the mucosal membrane, and it prevents the IgA from denaturation by bacterial proteases. J chain is a polypeptide that connects the 2 monomers, and 2 IgA subclasses have been described, IgA1 and IgA2. Then comes the IgM, the third most abundant one, and it constitutes 5 to 8% of the serum and occurs as a pentamer. J chain connects the dimer, and this is the first antibody to appear in circulation, which is intravascular. It is the largest and the oldest immunoglobulin, and this is the first immunoglobulin to be formed in the fetus, and it is not transported across the placenta. IgM is intravascular and is the principal component of primary antibody response. Presence of this IgM in fetus or newborn indicates infection. IgM deficiency is associated with septicemia, which is the multiplication and growth of bacteria in blood. IgM antibodies are short-lived, and hence their presence in serum indicate a recent infection. Its H chain is mu, and there are 4 main domains in the constant region: CH1, CH2, CH3 and CH4. A single molecule of IgM can bring about immune hemolysis, whereas 1000 IgG molecules are required for the same effect. IgM is 500 to 1000 times more effective than IgG in opsonization, 100 times more effective in bacterial action, and 20 times in bacterial agglutination. In neutralization of toxins and viruses, it is less effective than IgG. Then the fourth one is IgD, which resembles IgG structurally and exists as a monomer. Its H chain, otherwise heavy chain, is delta, and it's mostly intravascular and constitutes about 3 mg per 100 ml of serum and is importantly intravascular. It has a half-life of 3 days and it occurs on the surface of unstimulated B lymphocytes. Majority of the cell-bound IgD are of K-type, which is kappa, and serum-bound IgD are L-type, lambda. The last but not the least is the IgE, immunoglobulin E, which resembles IgG structurally because it exists as a monomer. It is found only in mammals and has a molecular weight of 1,90,000 and half-life period of 2 days. This is an extravascular immunoglobulin. Normal serum level contains only traces about a few nanograms per milliliter, but greatly elevated levels are seen in allergy conditions like asthma, hay fever, and eczema. IgE exhibits unique properties like heat solubility and get inactivated at 56 degrees Celsius for 1 hour, and affinity for the surface of tissue cells, mainly the mast cells of same species. When antigens attach to IgE receptors, they produce histamine. It neither causes complement fixation nor cross placental barrier, and Ig is chiefly produced in linings of respiratory and intestinal tracts. This is the overall idea of the types and classification of immunoglobulin that is present in the human serum, and this is how it works, this is how do they perform their function, and this is how do they work together to help in their proper working and getting the immune system and boosting up the immunity. And this is the idea of the video. Hope you find this effective. If you find this video helpful, please do like, share, and subscribe. And if you have any kind of doubts and other suggestions, please let me know through my comment section. And also, if you have any kind of other relatives or friends or any other people who are interested to learn biology, please do share this video to them also. And that's all for today. Thank you for watching. God bless. Thank you. See you in the next video.